Perfect. Yeah, I'll just start the introduction. Hi everyone, I hope you all are well. I'm Yi Xing, co-president of Biological Society. Thank you very much for joining us today on either Zoom or live on YouTube. We are very grateful to have Dr. Jing-Ke Weng as our speaker today. Thank you very much for accepting our invitation. We invited Dr. Weng upon the kind suggestion of one of our members, Hong Hao Su, a plant scientist going to his third year of undergraduate studies here at Cambridge, and he is a keen follower of Dr. Weng's research. He will be introducing Dr. Weng and also initiating the Q&A session, while Christoph, our co-president, would be moderating the rest of the Q&A session today. If you're on Zoom, feel free to post your questions or ask your questions in person towards the end of the talk. And if you're on YouTube, feel free to post your questions and comments on the live chat session. I shall now pass the floor to Hong Hao to introduce Dr. Weng.
Jing-Ke Weng as our speaker today. Dr. Jing-Ke Weng is an associate professor at MIT and a member of the Whitehead Institute. Dr. Weng's lab has a special interest in understanding the origin and evolution of metabolic pathways in plants and how plants utilize those metabolites to interact with the biotic and abiotic environment. In addition, Dr. Weng's lab is keen to utilize plants as a model system to understand disease caused by protein misfolding. Dr. Weng's work also aims to elucidate the mechanism that underlies the efficacy of traditional plant-based medicine, from Ji Hong's 1,600 years old prescription for emergencies to Yo-Yo Tu's Nobel Prize-winning work that built upon these ancient observations. Now let's welcome Dr. Jing-Ke Weng to give his talk.
Yes, thank you so much, everybody, Hong Hao and Yi Shen and Christoph for the very kind invitation. Before this seminar, the minutes before I was chatting with everybody, it's particularly challenging time for young people, for students. It's the same on this Cambridge. So since I'm at MIT, we've made plan that the fall semester will go online, mostly for the lectures. But in the same time, there are silver linings. So I think it's a downtime for everybody to kind of reflect. And one thing that came about of all that is biology or biomedicine are quite important disciplines. So you guys are in the right game. So I think there's bright future ahead of us, and we just need to work towards that. Okay, so today I'm going to share some of the work from my lab. So my lab, the research in my lab is pretty much inspired by the remarkable biodiversity that exists in nature. So since I was a kid, I was quite amazed about plants, insects in the park and backyards near my home. Many years later, I picked on biology. Eventually, I fell in love with biochemistry, and I realized there is another layer of diversity that's not really visible to your naked eye. So this is so-called the chemical diversity. So in each of the organisms that we analyze, particularly plants, they're filled with those small molecules, these chemicals that you can detect and analyze by instruments, but you cannot really see them directly. So plants are particularly good of making these molecules. One major reason is plants are known as so-called sessile organisms. So once they're rooted on the ground, they cannot move around easily. So whatever they do to deter herbivores, to avoid being eaten by insects, or to mate, or to avoid the stresses on the terrestrial environments, they have to do this through chemistry. So this is a very different strategy than animals which have evolved very advanced motor systems and nervous systems to actively seek for food, finding mates, or avoid danger. So to do that, over hundreds of million years, plants came about this remarkable metabolic systems to make all these metabolites. And I would say that this chemical space, the whole collection of the metabolites in nature is barely explored. So we only know the tip of the iceberg. So it'll take decades, if not hundreds of years, for us to continue discover the chemistry and the biochemistry behind this remarkable chemodiversity in nature. And for plants, these molecules are quite important for humans, and we consume them in our daily lives. We drink morning tea or coffee. We use spices for cooking. So we're actually utilizing the aroma or certain properties of the small molecules from plants for our daily use. And we also rely on plant natural products as energy and material. For example, the wood, which is lignin, and rubber we use to build a lot of materials are also coming from plant metabolism. And talking about biomedicine, the relationship of plant chemodiversity and medicine, in the early 20th century, actually most of the drugs approved by FDA are coming from plants. And so here are just some examples. On the top left is morphine. So this is the first FDA approved drug, and it's coming from the opium plant. And there are many first-line chemotherapy drugs that are coming from plants. So here are some examples: paclitaxel, vinblastine, which are tubulin inhibitors, and we also have homohairatonin, topotekans, and etoposide. So these are very complicated molecules with complicated structures, and these are very useful molecules for treating cancer. So on the bottom right, so we have huperzine A or icarisid. So these are very interesting molecules, but coming from plants that are almost endangered. So they have potential to become modern medicine, but they didn't because they have some issues. For example, the sustainability of getting those plants. So I'll comment on these issues later on. So one other aspect I would like to discuss why we need to study plant metabolism is to do with climate change. So this is a little bit of history. In the mid-19th century, human beings discovered that we could use very old plant natural products, essentially oil that's stored in the ground, and make that into an energy source. So that in the following century, so the whole society has become a society that totally rely on fossil fuel. So now the fossil fuel industry accounts for about 15% of the world's GDP. Not only our energy is almost entirely coming from the ground, many of the fine chemicals are also derived from this industry. So all the plastics we use in our daily lives, and medicines, and actually the pills, if these medicines are synthesized organically, the raw materials are actually also coming from fossil fuel industry. So this is kind of alarming because as we use more fossil fuel and actually the damage has been done, we have now very clear evidence supporting the global warming is very much tracked with how much CO2 and other greenhouse gases that are derived from human activities. And this rise is at a very alarming rate. And as a whole humanity, we got to do something. And there are signs showing we have to act right now. If we do not do it now, it might be too late. So this is on my mind when I do everything to do with plant metabolism, because the science and technology we learned how plants can fix carbon can eventually help us develop new technology to reverse climate change and global warming. So in the last 100 years, chemistry, the discipline of chemistry has really flourished. and organic chemists have developed ways to synthesize useful and elegant molecules. But I would argue that nature still offers greater complexity that till now humans cannot quite imitate. So here are some examples. On the left, you're looking at a glue. So essentially this polyvinyl acetate. So it's a polymer. When it dries, it can bond things. And in nature, there are other kinds of polymers which have much more complex structures. So this is a paper my lab published last year reporting the structure of the hardest polymer existing on Earth, the diamond of the plant world, the spore polymer. So this is the polymer that coats the cell wall of the pollen or spores of plants. And they have very remarkable structures contributing to their inertness and sturdiness. So we're far from understanding how good the plants can be as chemists. In the drug discovery field, although nowadays major pharmaceutical companies can all claim they can make chemistry very efficiently. Actually, each major pharmaceutical company all have chemical libraries that contain millions of compounds. But these libraries are usually created by high throughput parallel synthesis. So although the numbers are high, their chemical diversity or chemical structures are relatively limited. So this is a paper I cite here. So if you do principal component analysis based on chemical structures, you find the natural products have way more diverse chemical space than the complete synthetic drugs on the bottom right. So that's no wonder currently there's a very large fraction of the drugs we use in the clinics are either natural products or natural products derived. So here are some real examples. So Taxol on the left, it's a chemotherapy drug coming from yew tree. As you can tell, it's very complex. It has many chiral centers, structure is complex. And on the right, you have imatinib, so Gleevec. So this is a very famous anti-cancer drug. Essentially, it's a flat structure. It has no chiral centers. You can represent the whole structure on the two-dimensional space. So all the compounds I've been talking about actually belong to so-called specialized metabolism. So this term is in the title of my talk. And this is different from primary metabolism. That's probably more familiar to you. For undergraduate level biochemistry, we learn mostly the primary metabolic pathways, the pathways that are useful for making building blocks to support life or generating energy. So for example, the TCA cycle on the left is a primary metabolic pathway. And on the other hand, the specialized metabolism is not required for survival. So here is an example of Taxol biosynthesis from the yew tree. The pathway is very complicated, but if you knock out this pathway in the yew tree, the tree probably doesn't die immediately. But these pathways contribute to the fitness of their native hosts to live in their native environment. So eventually the tree population may decline. Maybe the Taxol is very important for defending a fungus pathogen. So over a long period of time, because of the fitness decline, these organisms may not survive, but these pathways are not immediately required for survival. And we also found these pathways distribute across the kingdoms of life taxonomically. So not all organisms contain those pathways. That's why they're called specialized. And these pathways really evolve driven by very harsh competition of these native plants living in their native environments. They're chewed upon by insects all the time. There are all kinds of microbial pathogens trying to live upon plants. And in the same time, the plants are trying to attract beneficial organisms like pollinating birds, insects, or beneficial microbes in the roots to fix nutrients for the plant. So these are the driving force for the evolution of these very complicated pathways. So over the years, the field has done quite a bit of research, and all summarizing these points of data points all together, we now have a good picture to capture how these pathways could have evolved throughout evolutionary time. So here, what I'm illustrating are different phylogenetic trees of enzyme families. So in the plant genomes, actually a big fraction of the genes are encoding enzymes. For example, the red ring captures the cytochrome P450s. So each plant genome contains about 200 to 300 P450 enzymes. They have very diverse roles in small molecule biosynthesis. So animals also have cytochrome P450s, but they're mostly responsible for detoxification of toxic chemicals. And these enzymes, they have really radiated, these enzyme family really radiated during plant evolution. And they're really borrowed from those enzymes that existed in primary metabolism. So here is one example. CHS is a plant specialized metabolic enzyme. It's derived from this Cas3 enzyme that's involved in fatty acid metabolism. As you can tell, the three-dimensional fold is essentially identical, but certain mutations have occurred that turning Cas3 into CHS, which becomes something that's very specialized, as you can see here. So that's one enzyme. And if you connect different enzymes together to form a pathway, and then you have a very specialized situation, because these evolutionary assembly of individual enzyme is a lineage specific event. So at the end of the day, in different species, if you have an A9 molecule, that's probably quite specialized, and you may only find this compound in a single species. So the other level, so if we dive a little deeper, we can look at the molecular details in each enzyme. So that kind of brings up the idea of enzyme promiscuity. So as we were taught at undergraduate level, we usually taught that enzymes are perfected catalysts. They're like perfect machines. They behave the way they're supposed to do. But if we analyze enzymes in detail, we found they're actually not so perfect. Sometimes they make mistakes. So as the top case here, this enzyme 1, most of the time is converting A to B, but sometimes it can also use the substrate C and make a compound D. So this happens at a very low level, so it doesn't really affect the normal function of the organism. But what if this organism encounters a new environment where D becomes beneficial? So then molecular evolution works to kick in some more mutations that D gets amplified. So for individuals that have more D will be selected. So then through events like gene duplication followed by further positive selection, then this ancestral latent function can now become a main function to create a new enzyme, E2. So if you think in a way that involves ancestral metabolic system, and these kind of events happen all the time, eventually ancestral pathways will give rise to new parts that can be assembled into a new pathway. So here's that was kind of the background of metabolic evolution, the way we view it. And now we're really living in a very exciting time when we're in the midst of genomic revolution. So when I started my graduate school, so that was just the beginning of it. So at the time, the first plant genome was sequenced, and also the first human genome was sequenced. The price tag for the first human genome was $3 billion, so essentially $1 per base. So that kind of provided the blueprint for many molecular geneticists to work for the coming years. In the midst of my PhD, so I was part of this phylogenetic genome project. That was the last genome that was sequenced by Sanger sequencing. The cost has gone down significantly. And last year, my lab published the high quality chromosome level assembly of the firefly genome. So that was done by the PacBio sequencing plus Illumina. So at a scale of tens of thousands of dollars. And we did this through my lab with some collaboration with colleagues. So it becomes very feasible to rapidly access genomic information from all kinds of organisms you're interested in. So that kind of leads to a new workflow that now allow us to study so-called non-model organisms. So not all plants are like Arabidopsis that can be easily grown in a lab or do genetic testing. Many plants of interest are trees. They're from environments not very much cultivatable in the lab. So over the years, we have developed this workflow that's really enabled by the genomic evolution. So when we have a plant, usually we do very fine sampling. We collect leaf, pollen, stem, roots, flower, different parts of flowers. And then we can process for both RNA sequencing. So that's to capture all the genes that are expressing a certain tissue. And then we also do metabolomics. So we capture all the metabolites in a tissue using mass spectrometry. So here is one example. So this is marijuana, which my lab doesn't study for legal reasons, but this makes a good example. So on the surface of the leaves, they have these structures called glandular trichomes. So these are very specialized cell types that contain or manufacture all the cannabinoids, which get people high. So when people sequence the transcriptomes of these tissue, they found the genes are loaded with cannabinoid biosynthetic genes. So then you can go on and use these candidate genes to test their function. And the way we test these functions, we actually do this through building. So this is using the synthetic biology tool, not just for engineering purposes, actually for learning purposes. So Richard Feynman said, I cannot understand what I cannot build. We really kind of took that quote in heart. And the way we do this is if we have a set of candidate genes we think are required for making a natural product of interest, if we can assemble these genes in the right order in another system, let's say in yeast or a different plant that doesn't produce this molecule, the time that you can produce this molecule, you have find the right set of genes. So you can actually learn by building. So this is opposite to learn by breaking. That's the traditional genetics when you mutagenize your population trying to identify mutants. When you can build, it's another alternative way of learning an unknown system. So when you do that, actually you have created a prototype that then you can further amplify to produce these molecules. So that leads to a new field called metabolic engineering. And in many different industries, as I mentioned, for biomedicine or commodity chemicals, this is a rising field. So for the audience who are thinking about career choices, this is going to be a very hot area in the years to come. So just to summarize this workflow, now we can discover natural products and utilize natural products from plants in a very different way. So we start with a non-model medicinal plants with interesting properties. We discover the genes that are responsible for making these products. And then we can put these genes together in a different system to recreate the product itself or to make derived products. So this is kind of the workflow we're thinking about. So over the past few years since I set up my lab at the Whitehead Institute, and we've done quite a bit of these exercises. So here is a slide summarizing. So on the top are the plants we studied, and at the bottom are the people in my lab who have done this work. And hopefully, it's a long process. So at this point, we figured out the chemistry and biochemistry, and hopefully in the years to come, we can turn these very interesting molecules from plants into medicines. But that takes a lot more effort from this point on. So today I'm going to focus on two stories just to illustrate this workflow I talked about. So the first story is a set of molecules coming from this plant called Kava. So it's a plant native to Polynesian islands. And I don't know how many of you have been onto those islands. If you go there, you will be, the local people will serve you the Kava drink. So it's really a high honor to do this. And this Kava drinking tradition has been with those native tribes for thousands of years. Through these drinking sessions, older generations pass their wisdom and tradition to the younger generation. And between tribes, that's how the conflicts were resolved. So Prince Charles and the Pope, when they visited Fiji years back, they also drank Kava. So this plant is only found on those islands, and it's in the Piper genus, and it's related to black pepper. So in the United States, Kava drinking is becoming more popular. So this is ESPN showing, reporting many athletes are taking Kava as a safe painkiller after their training. So it's not addictive and it's quite safe. So I heard about this story probably now 5 years ago when I attended a seminar at Harvard Museum. And as I was listening to this, I was quite amazed. So I took my phone and looked up this plant. It happens that the chemistry was pretty much worked out. So people knew that Kava, because they have this relaxing activity, that's because they contain this set of molecules known as kavalactones. So these are so-called type 3 polyketides, very unique to plants. And Kava has become very good at making it. So each plant can accumulate 20% of their dry weight as kavalactones. And it's quite safe for people who drink Kava. One day they could indose about 2 grams. So that's quite amazing. In addition to kavalactones, the plants also make some molecules known as flavokavenes related. So when I saw this, I immediately formed a biosynthetic hypothesis how these kavalactones could have been made. So before this, I was working on flavonoid metabolism. So for classical flavonoids, you actually start, as shown on the bottom, you start with an intermediate known as p-chromol-CoA, this one. And then you need a very conserved plant enzyme known as CHS, which I showed before, which is a type 3 polyketide synthase. Then you can extend this starting molecule as polyketides. In this case, the CHS will produce a tetraketide. That means there are 4 ketones. And this structure is high energy and not stable. And this will cyclize in the active site of the enzyme to make chacone. So eventually, this is the pathway leading to many flavonoids, including flavokavenes, as we see in Kava. So how do you make kavalactones? So by looking at tetraketide synthesis, what if this pathway is somehow shortened? Instead of making a tetraketide, you can make a triketide, as illustrated on the top. And through a different cyclization mechanism, then you can create a lactone ring. So this becomes the backbone, then you can derive into many different kind of kavalactones. So this was work done by a very talented postdoc in my lab, Tomasz Plaskel. And he took this hypothesis and was able to acquire multiple Kava plants from those islands mostly from Hawaii. And in the same time, we also analyzed the genes of related plants, those non-kavalactone producing piper plants. So when we built this phylogenetic tree of the type 3 polyketide synthase genes, this is just very similar to the tree that Charles Darwin built based on species trait. So you can also do this with genes. One thing we realized is that Kava plant, actually, in addition to containing the canonical CHS, also they contain two seemingly derived polyketide synthases. So we named them SPS1 and 2. And there's, when we do the positive selection test, we found this particular branch has gone through positive selection. And it's also quite interesting that these two genes are highly expressed in the roots where kavalactones are accumulated. So this piper nigrum is black pepper. And as you can see that this species doesn't contain SPS genes. So it seems very likely these derived polyketide syntheses evolved from CHSs likely are the polyketide syntheses and responsible for making kavalactones. So then we took these genes and then we expressed these in vitro and test their biochemical activities. And indeed, they are the kavalactone synthase. And so they're right. So that this particular experiment verified our initial hypothesis. They're very dedicated kavalactone synthase. Very interestingly, the original CHS in Kava can also make kavalactone. So that also kind of consistent with the hypothesis I presented previously, that the latent activity already existed in the ancestral copy of the SPSs. So we were interested to find out what molecular features dictate these unique activities in SPS1 and 2. In this case, we crystallized the Kava CHS and the Kava SPS1 and compared their structures in light of the sequence alignment. So we found there are some unique substitutions that are near the active site that have very subtle but important contribution to the shape of the active site. So particularly in SPS1, there is this insertion of threonine residue 194. So this insertion plus some of the substitutions actually shrink the active site volume by a little bit. So instead of being able to accommodate a tetraketide extension, as I showed before, now in SPS1 can only accommodate a triketide extension. So this explains how molecular evolution in the gene can eventually contribute the new functionalization of a particular enzyme. So very similarly, we were able to find additional enzymes. For example, after this polyketide formation, lactone formation, you need a stereo-specific reductase to convert this conjugated double bond into this chiral center. And we found this enzyme. And we also found methyltransferases that modify specific OHs to make methoxy groups. So in the same time, we also solved the structure of this reductase and showing the structural mechanism of stereo-specific reduction of this double bond, as shown here. We also found one P450 enzyme that's responsible for making this methylene dioxy bridge moiety here. So altogether, the discovery of these genes allow us to elucidate the kavalactone biosynthetic pathway. Then now we can explain the biosynthetic mechanisms for almost 20 different kavalactones that naturally occur in Kava plant. And we can produce 10 different kavalactones in the microbial system. And we're now adding into this system additional enzymes, for example, the bacterial halogenases. So now we can make so-called new to nature kavalactone analogs. So here is some fun fact of this project. This is Tomasz. When he started the project, he bought some kava from Amazon and had his very first kava tasting with his wife at home. And it looks like milkshake, but actually tastes pretty horrible, very bitter and peppery taste. But the next morning he reported it actually worked. He said that it's very sedative, and he actually felt happy. And since then, we've hosted several open public kava tasting events at Whitehead Institute, which has been very popular. Yeah, myself have tested multiple times, and I can also testify it really works. So the point to illustrate here is many medicinal plants have long time of traditional use. They are effective based on people's experience, a lot of time anecdotal, but systematic. And they are safe. Definitely there are plants that are not safe, but there are cases where there are quite safe herbal remedies for certain conditions. So then we want to take that data and to study the mechanism. Hopefully we can bring a new medicine that can go through clinical trials. So in the lab, we use a zebrafish system. So we found zebrafish is a very easy to use vertebrate system to test pharmacology of natural products. So in this case, if you add kava extract into the fish water, and as you can see, the fish will have trouble maintaining balance, and then it's kind of you can really see this is happening, because if you drink kava, you also want to sit down because you feel a little dizzy. But you can retrieve the fish, put in normal water, it'll recover pretty quickly. And we can do quantitative analysis of fish behavior. So this is a test known as signal taxis. So when fish is anxious, it actually tends to swim around a container. So in this case, we're using 24 well plates. In each well, we place a little fish there, and then we can record the fish behavior through a video camera and quantitate the time they spend in the outer zone versus the inner zone, and then do quantitation. So here is the real data. When fish is swimming around the well, as you can see here, that means the fish is quite anxious, versus if fish is doing something here in the bottom left, they're relatively calm. So when we quantify these, we found all kavalactones, 6 individual kavalactones we tested, all reduced fish anxiety. Whereas as controls, flavor carvings and caffeines have no effect. So we are trying to identify the best molecules which we can then move into other animal studies, hopefully making one of them a new drug for treating anxiety. So in the same time, we're quite interested in how different kavalactones interact with receptors in human body. So there have been a body of literature in this space. For example, people found that most of the kavalactones are GABA receptor antagonists. And there's one of the kavalactones, yangonin, as I shown in the previous slide, is also binds to the cannabinoid receptor. So that's why when people drink kava that have different combination of different kavalactones, they actually have different neurological responses. So we found this is quite intriguing if there's a single molecule that can modulate multiple neurological receptors. So these have great potential moving forward as neurological drugs. As we know, opioid epidemic is really a problem in the US and in the world. And we're really hoping that, inspired by traditional medicine from different indigenous cultures around the world, we can come up with new medicine for treating these conditions. In the rest of time today, I want to talk about a different system. So this is a little bit different from kavalactone and illustrate something I think really can be harnessed through genomics. So these compounds, as shown here, are known as lyciumins. So these are quite specialized, so-called branched cyclic peptides that you only find in plants. And in some certain classes of plants, for example, goji berry. So in traditional Chinese medicine, the roots of goji berry have been used for treating hypertension. And people identified these molecules from the goji berry roots, and they found these compounds are very potent inhibitor of angiotensin converting enzyme, ACE enzyme, and that explains why the goji berry root can have efficacy treating hypertension. And very interestingly, if you are following the COVID-19 situation, you all know now ACE is also the receptor for the virus. So potentially goji berry and these molecules we're looking at now can also be used for fighting COVID. So these molecules are quite unique because of two structural features. One is, as shown here, this is called pyroglutamate. So it's essentially a glutamine that's self-cyclized. And the second structure is this ring that's formed by nitrogen carbon bond formed between an indonitrogen coming from a tryptophan with a backbone alanine. So these two structural features making these molecules super stable because there are no obvious enzymes can degrade these structural features in these compounds. So the work I'll produce later are done by a very talented postdoc, Roland Kirsten, as shown here. So the peptide natural products in bacteria are known to be produced through two very distinct mechanisms. The first mechanism is through the so-called non-ribosomal synthetase. So it's a gigantic molecular machinery that produces very complicated natural products and through its own machinery. So the other type is known as RIPS, or ribosomally synthesized and post-translationally modified peptides. So these peptides are derived from ribosome. So through typical protein synthesis, the way it's done is you have a gene encoding a precursor peptide, and within the precursor peptide, you can find the core peptide. And before the core peptide is released, it's usually post-translationally modified by additional enzyme. And usually for cyclic peptide, the cyclization happens during that step. Then through proteolysis, and then you can release the mature peptide. So plants do not have non-ribosomal peptide synthetase. So actually most of the peptides known in plants are produced through the RIPS pathway. So, so when we know this case, we know goji berry produced lyciumins. The first thing we did was to get the plant and assembled a transcriptome from the roots. We know these lyciumins are highly enriched in roots. And from that transcriptome, we identified a potential precursor gene. And it's very hard to miss. So this is a highly expressed transcript that ranked number 13 out of 13,000 genes. And it carries a burp domain. So this blue part is a burp domain of unknown function. And the N-terminal end of this gene actually have many repeats of the motif that are seen in lyciumin chemistry. And the most remarkable thing is when we clone this one gene and then transiently expressed in the tobacco plant. So this is a very nice species, Nicotiana benthamiana, that many plant biologists use in the lab for transient expression. So we can put this gene in agrobacterium and then inject the agrobacterium soup into the leaf interspace. And within days, then you can analyze the gene product of that experiment. So by just expressing this one gene, the Nicotiana plant can actually produce all three lyciumin products in its leaf. Nicotiana is not known for producing branched cyclic peptides, but just producing this one gene, you can actually get to the products, indicating all the other components necessary for producing this molecule is probably contained in Nicotiana. The only missing component is this precursor peptide. So by this first identification, that really allowed a new workflow to discover new natural product chemistry through genome mining. So this has a lot of advantage, because for natural product chemistry discovery, usually you start through starting from a lot of the plants and do extraction, isolation, then identify the structure. But now we can really read the genetic information from the sequenced genomes or transcriptome, and then predict what kind of chemistry these plants can produce. So this is really enabled because it's initial discovery. So we did that. We looked into 116 genomes that are available on NCBI, and we actually found 21 of them contain lyciumin precursor peptides. And these plants are mostly in the amaranth, the beans and solanaceae families. And here are some examples. So most of these are either grains or vegetables. For example, the amaranth family, including quinoa and amaranthus. So these are all very important grain plants. And also we have the soybean and medicago plants also produce lyciumins. And very interestingly, in the solanaceae family, you have the cheap russet potato and the more expensive red potatoes. And we analyze these compounds are produced in the buds, potato buds. So there are good reasons you should not eat potato that are budding, because not only to some other toxic compounds, they also contain lyciumins, which could potentially harm you. Yeah, and they actually produce different chemistry. That's quite interesting, that these genes could readily evolve just within a very short period of time. It can result into chemical divergence here. So from these naturally occurring lyciumin cases, now we can summarize the structural rules for this peptide. And we found that in the R1 position here, it's usually aromatic residues, and the R2, R3 are more variable. And there are certain positions you cannot change. For example, this tryptophan and this glutamine has to be the way this is. And we also categorize these peptides into type 1 and type 2 lyciumin peptides based on where the core peptide is relative to the birth domain. If it's embedded, we call it type 1. Sometimes it's out of the birth domain, then we call it type 2. And we also surveyed across different plants. Most surprisingly, we found lyciumin type of peptides also exist in Selaginella. So for people who know plant taxonomy, Selaginella is a sister group of lycophytes. So these are basal vesicle plants that have diverged from all the flowering plants 400 million years ago. So very ancient plant, and they seem to have evolved lyciumins on their own. So it's a case of independent evolution. And their structures, their lyciumin structures are also slightly different. So the indole nitrogen actually is bonded with a serine instead of a glycine. Yeah, so when we surveyed this, we found there are lyciumins are present at least in 5 different plant families. And most of them have evolved independently. The diversity came about independently. But very interestingly, out of all these lyciumin diversity, we found there are 2 lyciumins, lyciumin J and A, are shared by multiple plant families. So these are so-called parallel evolution. Some people call them convergent evolution. I don't use this term for this case because their ancestral states are quite similar, so I call them parallel. The fact that different, very distant plants that evolve the same chemistry, indicating these compounds must be very important for the plant physiology. We're still in the process trying to understand how they work the way they are. So we are also in the process understanding its biosynthesis. We know the cyclization happens first, and this enzyme is very likely to be actually the burp domain itself. We have some evidence showing this. There are endopeptidases and exopeptidases that do different cleavage. We also identified the glutamine cyclotransferase that makes this glutamate moiety here. We also tested the structure-function relationship of this peptide by varying the sequence of different residues. So we found there are 5 residues that can be changed into other amino acids, whereas 3 residues you cannot change them. And by doing so, we can create a library of over 3 million molecules. So it's just by engineering this one gene. So this is remarkably powerful to generate peptide chemical diversities that you can feed into drug discovery. We also found by varying the repeat numbers from 1 to 10, you can significantly increase the final yield of the peptide when you transiently express this gene in Nico channel. So this is a lot more than the native host can produce. So number 4 is Goji berry itself. It produces tiny little amount of lyciumins, whereas we can produce a lot more of the compound in transgenic plants. So this offers a new path for utilizing these molecules as drug. So just to summarize the system my lab is interested in, it's really facilitated by some of the recent advances in the areas of genomics, our fundamental knowledge about evolution of specialized metabolism, and tools from synthetic biology and metabolic engineering, and some other transformative analytical chemistry techniques. So by using this, we now can use the approach of combinatorial biosynthesis to not only create natural products themselves, but also in additional natural products when you combine genes from different plants and even fungi and bacteria. So that can then feed into pharmaceutical industry, nutraceutical, and commodity chemicals, including energy. So I think it's a very exciting field, and it's really barely explored. So if you guys are interested, I highly encourage you guys to look into this area. So at last, I want to thank people in my lab. We have a very nice group. This picture was taken last year. We're doing our summer hiking. Too bad it's not happening this year. To particular scientists, Roland Kirsten, Tomasz Plaskel, did the lyciumin work and the kavalactone work. And both of them have recently started their own lab, one in Czech Republic, one in University of Michigan. So, and other people in my lab also contributed to the work. So that's pretty much for my talk today. I'm happy to take any questions you may have. Thank you.
So thank you for that fascinating talk. And if I can open up the floor to questions from the Zoom group chat. And also, of course, if anyone would like to ask questions on the YouTube live chat, I'll be monitoring that and passing the questions on to the speaker. So there's anyone, if you have a question on the Zoom chat, please speak up. Perhaps if I start off with a question, regarding the non-ribosomal synthetase that you can find in bacteria, do you think that perhaps would be beneficial to maybe introduce that into plants? Do you think that there could be something to it, or do you think that's not really a...
Yeah, that's a very interesting idea. There are a lot of people who are interested in engineering NRPS, non-ribosomal peptide synthetase. it's quite challenging, even just to engineer them across microbial species. So the way microbial genes work, that they actually function in gene clusters. In bacteria in particular, they function as operons. So there are many elements encoded on these gene clusters that need the host, the native host, to work. I'm not saying it's not feasible to engineer them into plants, but there will be a lot more learning to do. Essentially, we actually do not know the full picture, how different elements on an NRPS gene cluster are regulated. So that's challenging, but there is one case of NRPS in Drosophila. So that means there has been one particular case where NRPS somehow evolved or transferred from microbial genome to a eukaryotic genome. So there is some evidence that this is feasible, but I would say it's quite challenging at this point.
Thank you. Any more questions from live chat or from Zoom?
There's a question about learning by building versus learning by breaking. Yeah, so this is a kind of idea I kind of summarized over a few years of practicing, because when people talk about synthetic biology, some primary researchers would say, oh, this is a little bit secondary, right? We are doing the prime work of discovery, and these molecular engineers will just build upon what we learned and try to build things. But what I really figured out, it's not only an engineering tool, but can really be used for learning. So I mentioned this Nicotiana benthamiana transient expression system, where you can take genes from other plants and quickly test their function in the tobacco plant. So what we realized is not only you can express one gene, you can actually combine multiple genes together to test their functions all together in a plant cell, or very much like an in vivo system. So one of our latest studies, we co-expressed 30 genes from a different organism in Nicotiana and found the product is readily made. So we have very little assumptions about our hypothesis. We think, okay, these genes might be P450s, but there are so many P450s in the native plants, so we have no idea. So what we did is we just cloned all 30 most highly expressed genes and then co-expressing Nicotiana, and the final product is there. Then we narrowed down our gene list, eventually found only 2 genes are necessary. So as the tool is progressing, I think we'll have much more power to use this approach for learning. So genetics, mutagenesis, I think, has a lot of limitation. For genomes that are big, for genes that are redundant, you actually run into a wall. There are certain hurdles, it's just very hard to overcome. So I would say the synthetic biology offers an alternative and complementary approach towards gene and function discovery.
Thank you for that question. One more question from me. Regarding the model systems in which various proteins and expression systems could be inserted into, I've read in your paper that, in your review, that the best way of, currently one of the best ways of inserting entire systems is into tobacco cell culture, I think. Do you think that that's just a property of the fact that tobacco has been a useful model plant for us for a number of generations, or do you think that there will be new and more efficient systems that will be far easier to use, for example, with algae?
Yeah, so my lab has not worked extensively in algae. Definitely algae is an attractive system because you can scale up the production using fermenter. I think there's a lot of historical reasons why tobacco plant is utilized so often. And once something works, I think researchers are not very much willing to switch system. So my lab is actually trying to explore additional systems, not necessarily model systems. But what we realized that the reason why tobacco plant is so great for transient expression, because their leaves are just very easy to be infiltrated. This is a very practical reason. Maybe the stomata are just bigger. The air, those pores for uptaking CO2s, maybe they're more densely populated on the leaf surface, and they're just easier for the liquid to penetrate through. We found some other plants, just some random plants we're interested in. You can also infiltrate. When you do that, you can reassemble pathways also. I think microbials, bacteria, and the fungal species have a lot of potential. We use a lot of Cerevisiae yeast. We're also moving to Pichia yeast. So yeast provides a eukaryotic cellular environment, so where many enzymes can fold and assemble. If you can do this in bacteria, it also offers some certain benefits. So I think researchers should all explore all possible so-called chassis organisms to express pathways of their interest.
Thank you for that. Any more questions from live chat or from Zoom?
Yeah, I actually have just like a general question, because I'm personally quite interested in, quite fascinated by the effects of the potent effects of some traditional Chinese medicines and herbs. Are you currently, could you elaborate a bit more about the traditional Chinese herbs that your lab is studying, perhaps now or in the past? Thank you.
Yeah, so it's there's a very thick body of studies that essentially accumulated across civilizations across the globe and through thousands of years. So I think I'm, essentially facing a mountain of knowledge and just starting. I think the first baby step. So it's reading into this literature is a very humbling experience. So currently, my lab is quite interested in some work which was mentioned during my introduction by Zhang Zhongjing. So he lived over 1,000 years ago. During time, there was a lot of wars and disease, war-related disease. When there's war raging, people are dying, get injured, and so the cold damage essentially is bacterial, viral infection. Some of the documentation saying basically the pandemic lasted for years, and at the time, without any medicine and understanding, it killed 90% of the population at the time. So that provided a very fertile ground for those ancient doctors to practice, because otherwise people just die. Why not just try different herbs, just see what happens? So I think some of the classical work, although there are a lot of things we cannot understand, they're very good starting points. So looking at the COVID-19, I wrote a recent commentary on this topic. So in China, Chinese doctors also prescribe herbal remedies for their patients, which contribute to lower death rate among severe COVID patients. I've looked into the recipes. So these plants have well-documented activity against viral infection, for example, a modulating immune system. So it definitely offers advantage over a single agent therapy. Things like COVID, you have lung failure, you have viral replication, you have your cytokine storm. So there are many complicated things all working together. So if you have something that's also complicated treatment, then you may offer some advantage. Although the science behind those kind of traditional treatment is very thin. So the currently we're basically looking to each individual herbs and their chemical principles and trying to discern their molecular mechanisms, how these molecules interact with their targets in human cells. And hopefully that's as I said, it's a baby step. In the future years, we can build upon this and develop multi-component systematic therapy that's inspired by those traditional herbal remedies.
Thank you.
And we have a question from the live feed about the kavalactone mechanisms. Could you possibly go into how the antagonism of the GABA receptor by the kavalactones give them anxiolytic properties?
Yeah, so many of you might have heard about this drug called benzodiazepines. So for people who have sleeping problems, and you'll be subscribed for that. So it's a very important molecule. So if that receptor is antagonized, and then you will go to sleep, essentially. But benzodiazepine has its own problem. So if you overdose on that, you may die. And there are some side effects. So some very interesting kind of area is this neurological diseases. Even the targets are limited. Let's say just this GABA receptor. If you modulate in a slightly different ways, you could gain a little bit different effect. And from our research, we actually found that kavalactones bind in a different site than the benzodiazepine. So you can treat the fish on benzodiazepine. Then on top of that, adding kavalactone, we found it has additional effects. So we also have biochemical evidence that the binding sites are different. So the binding kind of mode and also pharmacokinetics of the molecule, even between molecules and with the same target, would give people different neurological experience. So that's why we found these are quite amazing. And if you have a single molecule not only touching this GABA receptor, also in the same time cannabinoid receptor, that will give you a very different experience. So I think this is a very rich area we're quite interested in, and hopefully we can find the right molecule that's safe and will be efficacious for treating very specific conditions that people really need.
Thank you. Do you have any more questions from the Zoom chat or from live stream? So maybe if I could just ask about, of course, as you've discussed throughout this talk, there are a lot of differences between plants in terms of diversity of their metabolome. But in terms of the folding system, do you think that, or has it been shown that different plant species will have relatively similar folding systems for proteins, so that we can more or less insert genes into various plants and expect the same, perhaps not level exactly of expression, but the same folding?
Yeah, so protein folding is a very interesting topic to my lab. One kind of anecdotal evidence for that is many other organisms, so humans for sure, we have various versions of neurodegenerative diseases. So these are caused by protein misfolding. In yeast, people also found protein misfolding aggregation will cause pathology, but sometimes can be also beneficial. Similar mechanisms or similar phenomenon have not been really observed in plants. So we hypothesized maybe plants have elevated folding capability since they oftentimes live in very harsh environment. Or there could be other factors. Metabolites themselves could assist folding of certain proteins. And part of that is many of the molecules that people found to change misfolding kinetics or trajectory are plant natural products. So we're very also in the early stage trying to figure out the role of metabolism in protein folding. That's one. The second area that was quite new is recent years people found there are membraneless liquid-liquid phase separation. So essentially, these are membraneless organelles that are driven by phase separation of biological molecules. Could be proteins and also could be RNA nucleic acids. And we also think metabolites play a role in the formation and the disintegration of these organelles. Some of these liquid-liquid phase separated organelles may play a role in enriching metabolism in the cell. So this is an area that my lab is quite actively investigating right now.
Thank you for that answer. And I think we have one more question from Zoom chat.
Hello, Dr. Weng. So basically, I have read your commentary from the molecular plant briefly, and you mentioned about the long cleansing and detoxifying compound. So I'm just wondering, like, if it is possible or if it is best to elucidate the mechanism of how does the LCDC work, and what are the best ways to utilize the essential compounds in these the essential molecules inside this compound for the world to use in order to treat the coronavirus?
Yeah, that's a great and relevant question. So after this paper was published, then we got a lot of feedbacks. And I was actually commenting on the challenge moving a traditional herbal remedy forward. So in China, most of the people grew up with the culture. So I think there's very little barrier for the patients if the patients are dying, and then the hospital after experiences say this may help, and the patients have very high acceptance for these. And when the efficacy was tracked, so this was this recipe LCDD that was trialed locally and then broadcasted nationally. So there are thousands of cases that have been reported and has remarkable efficacy. The problem for that is it doesn't go beyond the country, because when there's a remarkable successful story, but then the other country never had the herbal remedy system. They don't know how to get it, how to prepare this, what's in it, whether, and for the regulatory agency to actually okay this. So there are so many hurdles that essentially limit even this successful one place for this to be then used somewhere else. So the only solution to that is to research, although the timeline will be very long. So as you know that the LCDD has maybe 20 different plants, and to know all the chemical composition of each ingredient and knowing all the mechanism for each compound and their roles to play in stop COVID, it's a very long process, if not decades. So it'll take some time, but you have to start somewhere. So there are some indication of certain molecules. For example, there's one cyclic peptide from Aston. It's been shown to have very potent antiviral activity. And there are certain compounds that are immunomodulatory. So even just these two, the combination of these two, I think it's a great combo for COVID. You need to stop viral replication. In the same time, you want to tame the immune system so that it doesn't overreact and kill your own tissue. So I think there are many good indications on how to treat this disease. Even we don't use this, we can learn from these kind of therapy and derive different therapies for coping with the COVID-19. But the challenge is the timeline would be long. So all the clinical trials currently are ongoing are basically repurposing the drugs that have been approved for a different purpose. Because the whole process, if you start from some new molecule, it'll be very likely 10 years before you can get it approved. By the time COVID-19 probably is gone. So that's the challenge, but we have to start from somewhere.
And thank you. And one last bit from me is like you also mentioned about like a little bit about social aspect of this issue. Like, how do you think like what the society's attitude toward plant sciences and herbal like the medicine derived from the traditional herbal ways of curing disease, like how the society should change the attitude toward utilizing plant sciences to treat diseases or to prevent the spread of further other diseases that could occur potentially in the future? Thank you.
Yeah, that's a great question. I think my stand on this is quite simple. It has to be science and evidence based for kind of a therapy to eventually be approved. It doesn't have to be fully understood, but it has to show, based on very high bar, it has to be safe and efficacious. So the standard set up by US FDA, which I think is very much adopted worldwide, is that you have to show something, whatever that is. It could be a mineral, it could be a plant extract. But if you claim that this can be used to treat certain disease, you need to show it's safe and it's efficacious through clinical trials. So I think we just work on this principle. And it can be from plants, it can be a single molecule, it could be a natural product, it can be a natural product inspired synthetic molecule, whatever that is. But we have to go, past these two bars, the safety and efficacy for something to really help patients.
Thank you. In that case, if there are no more questions, I think I'll just wrap up in that case. So Dr. Weng, just thank you so much for your time, for that fascinating talk in which you've introduced us to the process that your lab works with to plant specialized metabolism, its importance both in terms of its potential social impacts, even its potential impact currently of the COVID situation with the ACE enzyme, which acts as a COVID receptor, and also delineating some of the experimental methods that your lab uses to find out more about biosynthetic pathways, using the examples of kavalactone and goji berries. So again, thank you so much, and thank you to everyone for tuning in, for asking questions. And just a reminder to subscribe our channel and follow us on Facebook. We will have another talk on Saturday at 2 p.m. from our two student speakers, and we will also have another talk this coming Monday. So once again, thank you very much, and hopefully I'll see you, well, still this week.
Thank you. Bye-bye.